Have you ever looked down in your leg or arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later, it's gone. It's all healed. And you don't even think of anything of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice that they're a lot longer than the last time you cut them. Or again, let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. Now, what do all these things have in common? One major thing they have in common is mitosis. Now, mitosis is a type of cell division done by most of our body cells, and it's really important for your cells to divide. If they didn't divide, you wouldn't grow. So, how do you grow if you can't make more cells, right? So, one reason why you're bigger than you were in 5 years old is mitosis. Now, mitosis is also great for repair of damage. If you have some kind of accident, like when we are talking about that cut in your leg or in your arm, well, you want to make sure that it gets repaired well. So you have to make more cells to do that. Mitosis is really great for that. Now, it is really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells because that's something different called meiosis. Now, mitosis is done to produce body cells. And mitosis makes identical cells. And that is the main goal in mitosis, to create identical cells. So if you're trying to make more skin cells to replace worn out or damaged cells, you want to make sure you have identical cells replacing what was lost. So, mitosis makes identical cells. Like this one. Now, what the cell is actually doing most of its daily life, which is actually a phase called interphase, where in interphase, the cells are growing and replicating its DNA and carrying out its daily cell functions. That's where the cells spend most of their time in respect to the whole cell cycle. Now, mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. And we will discuss now the different stages in mitosis. We have prophase, metaphase, anaphase, and telophase. So I like to call these stages of division in mitosis as PMAT. P for prophase, M for metaphase, A for anaphase, and T for telophase. Now, once all is finished, we end up with two identical cells, each with all the genetic information pertaining to that organism. But let's start at the beginning and learn what happens each phase. So before mitosis begins, when the cycle is still in the G2 phase of the cell cycle, we have two copies of all the chromosomes sitting in the nucleus, but they are loose and strewn about. And after that, we will now discuss the first stage of mitosis, which is prophase. Pro means before. Now, there are two pairs of centrioles. Then, as mitosis begins, during the prophase, the chromatin becomes tightly coiled and forms the shape we are familiar with, which is the chromosomes, with sister chromatids linked by a centromere. It is also in the prophase that something called mitotic spindle begins to form. This is made up by two centrosomes and a number of microtubules that begin to form between them. Each centrosome has a radial array of microtubules surrounding it called an aster. As the cytoskeleton disassembles, the microtubules begin the centrosome grow and grow, which pushes them apart. And next to prophase, we have metaphase or the pro-metaphase, M for middle. Then We have, again, the prometaphase. The nucleus breaks apart, and the growing microtubules cover the area where the nucleus used to be. So that they can attach to special proteins called kinetochores, which have assembled on the chromosomes at their centromeres. Now things are starting to get organized as a kind of tug-of-war plays out. Then, in the metaphase, chromosomes have settled at the poles of each cell, with the aster attaching to the plasma membrane, and all of the chromosomes have been arranged nicely along a plane in the middle of the cell, and this imaginary plane is called the metaphase plate. At this stage, there is a checkpoint to ensure that each pair of sister chromatids is firmly attached to the opposite ends in the mitotic spindle. Once all the kinetochores are attached to the spindle and everything is lined up nicely, a regulatory protein complex becomes activated, allowing the cell to pass through the M checkpoint, which means we're now ready for anaphase. Anaphase means away. Now, in this phase, it is the shortest of all the phases. We have anaphase. Now, the enzyme separase, ito siya, cleaves the cohesins that keep the sister chromatids together. And the kinetochores attached to the sister chromatid pull the chromatids apart on each chromosome. So it is pulling apart, thus generating the two separate sets of the genome. These chromosomes are then pulled by motor proteins that attach to kinetochores, which reel them in by their centromeres to opposite ends of the cell. And with the microtubules, they are attached to, coming apart as they go. So they are now separating from one another. And the cell elongates during this phase until the two sets of chromosomes are far apart. Now, those are the stages in mitosis. First, we have the prophase, metaphase, anaphase, and telophase. Last, we have the telophase. Now, in telophase, two nuclei form, rebuilt from fragments of the original nucleus that came apart in the pro-metaphase. Then the chromosomes loosen up a little and the microtubule finish coming apart, and last, mitosis is complete with two genetically identical nuclei. To finish things up, cytokinesis will occur, which is where the cytoplasm, which has already begun dividing the cell into smaller ones, will continue until the cells are distinct and separate. This starts with a cleavage furrow at the metaphase plate, caused by actin filaments that pull the cell inwards like a drawstring, which eventually pinches the cell into two. Now, our body is constantly producing new cells by mitosis to make new skin, heal a wound, or when you grow rapidly in childhood. And also in mitosis, it is good also for growth and repair of cells. And also in cancer treatment. Now, how do cells know when to divide? Cells regulate their division by communicating with each other using chemical signals from special proteins called cyclins. These signals act like switches to tell cells when to start dividing and later when to stop dividing. Now, in cells, they also have this on and off switch. Now, what will happen if the cells will continue to divide? Or is it important for us or for our cells to stop? The answer is yes. There should be a stop or a stop process in cells or in mitosis. And if there is a continuous mitosis process, we can now have this cancer. Cancer refers to a group of diseases characterized by uncontrolled and abnormal cell division. And cancer cells form tumor, like the benign tumor and the malignant tumor. When I say benign tumor, it remains clustered together, but still they are replicating, or they are still growing cells or cancer cells. And next we have the malignant tumor, the tumor that can break away and metastasize. So it is growing rapidly. Now, example, we have the HeLa cells. We have the cells of Henrietta Lacks, who died of cancer in 1951 at the age of 31. But the cells of Henrietta are still growing in laboratories. That's why they considered, or they called the cells of Henrietta as the immortal cells. So in 1952, HeLa cells became the first human cell line that could grow and divide endlessly in a laboratory, leading scientists to label these cells immortal because they are still growing. They are replicating. The immortality of HeLa cells contributed to the adoption across the world as the human cell line of choice for biomedical research. So because of the cells, now because of the cells of Henrietta, scientists now can check, or can check some irregularities, or some or new discoveries from the cells, or from the HeLa cells. And that's it for the mitosis phases. Thank you for listening. Now we're going to compare and contrast mitosis to meiosis. On the left, we will have the diploid cells with 46 pieces of DNA, and the cells start and end in diploid. On the right, these cells will start with 46 pieces of DNA diploid. And it will go through meiosis to make new cells, but then there's a second round of meiosis. Now these two cells divide again to make 4 cells. Each of them have now half the amount of DNA. So I want to show on the animation on this presentation that follows this process a little more visually. Now we will check this out. Now, in the left, we're going to have a cell that will undergo mitosis. On the right, a cell will undergo meiosis. Before we get started, I just want to make a count. There are 4 strands of DNA called chromatin, loose linear strands of DNA in this animated cell on the left. And on the animated side on the right as well is going to have 4 strands of DNA or chromatin to start. Take note of it, and we will gonna compare that until the end. Now this key right here is gonna help us visualize where the DNA came from, where the chromosomes and chromatin came from. The red strands are our DNA that were inherited from the maternal side of the family from your mother, and the black strand, or the black strands of our DNA and chromosomes inherited from the paternal or to the father side of the family. So we have here the cell on the left is an interphase, the S stage of the interphase, and this is prior to mitosis. And what happens here is chromatin is duplicated, so the 4 strands have duplicated the DNA of chromatin were duplicated and become 8. So we have here the duplication of DNA strands. On the right, the same thing happened. The chromatin is duplicated so far, the cells really, you can't tell the difference between the two processes. Now, as we move on into prophase of mitosis on the left, the nucleus will dissolve. And in prophase 1, in meiosis on the right, the same thing will happen. The nucleus dissolves so far, and there is no distinction between the two processes. And as we move on to prophase of mitosis on the left, we're going to see the loose linear versions of DNA called chromatin coil into these X-shaped structures called chromosomes. And on the right, the same thing. The chromatin calls the chromosomes, and now we will number the chromosomes. And please notice how there are two chromosomes, number one. And one in red, and here one in red that you inherited from your mother, and black you inherited from your father. Same thing there. Same thing happened here. There's two chromosome number 2s. Now in humans, there's 46 chromosomes. So these two chromosomes, the 12 and two chromosomes 21s and two chromosomes 19s. For simplicity, I presented here a cell with 4 total chromosomes. And as we go on into prophase 1 of meiosis, what happens is synapsis. And this is where the two chromosome number 1s pulled next to one another by spindle fibers, and two chromosomes, and we have here two chromosomes number 2s have pulled next to one another. This creates groupings called tetrads. Notice how the tetrads are made from two chromosomes, number 1s, or two chromosomes number 2s. You will not see a chromosome number 1 paired with chromosome number 2. You would not see also a chromosome number 13 paired with chromosome number 19. So they are paired based on their numbers. So tetrads are created from the homologous chromosomes, and we have two chromosomes 12s and two chromosomes number 19s and two chromosomes number 2s. So these are what tetrads are. So why does synapses happen? Synapses sets up what's called crossing over, the recombining of the homologous chromosomes, and what happens is that portions of chromosome 2 in red will actually break off and reattach with portions of chromosome 2 and black. This recombines portions of our homologous chromosomes with one another. Now the same thing happens in the red portions of chromosome number 1 will break off and reattach with portions of the other number 1. So the tetrads experience crossing over. Synapsis and crossing over only happened in prophase 1 of meiosis, and this does not occur in mitosis. So we're ready to move on into the metaphase stage. Now what happens in metaphase stage? On the left, the metaphase of mitosis, spindle fibers will actually pull these chromosomes just randomly to the equator or to the middle line of the cell. On the right-hand side, spindle fibers will pull these tetrads randomly to the equator or to the middle line of the cell. And as we move on into anaphase of mitosis on the left, the spindle fibers will engage in a tug of war, where they pull on the chromosomes where the actual chromatids are pulled to the opposite ends of the cell. On the right side, anaphase 1 of meiosis, the spindle fibers engage in a tug of war, the same with mitosis, and actually pull apart the tetrads. The tetrads are separated with one chromosome being pulled to the left and one chromosome of a tetrad being pulled to the right. And so we'll move on into telophase. First, in mitosis, in cytokinesis, the cytoplasm begins to split the cell into halves. The nucleus begins to regrow, and as that happens, the chromatids begin to unwind back into their loose linear version of DNA known as chromatins. Now, on the right side, in telophase 1 of meiosis, cytokinesis again begins to split the cytoplasm in half to create two cells, and sometimes the nucleus regrows, sometimes it doesn't. Now, in this animation, I chose the regrowing of the nucleus. And if we evaluate the mitosis cell on the left, this is the end of the end result we've created, two diploid cells. Now, we're not finished with the meiosis on the right, so we're gonna end the description of meiosis here. Since our topic for this lecture is all about mitosis. It's all about mitosis. Now, in the meiosis result, notice that it's not just 4 cells that are created, but we have 4 cells that are haploid. There are 4 haploid cells, but in mitosis, we have 2 diploid cells. Now, the cell on the left, the new cells have the same amount of chromatin as we started. Now, take note that there are 4 strands of DNA in the start, and that is our example here. Now, and notice how these 4 strands of DNA in the 2 cells created by mitosis. So still we have 1, 2, 3, 4, 1, 2, 3, 4. So it created 2 diploid cells. Now, on the right, I noted that there are 4 strands of DNA for our example to start, but the new cells only have the half the amount, and they only have 2 strands of DNA. This is because of the second round of meiosis, we created haploid cells. And in mitosis, the cells created are identical. There's 2 full strands of black, 2 full strands of red chromatin. But the cells in meiosis are genetically unique because of crossing over and recombining of those portions of homologous chromosome way back in prophase number 1. So they are genetically unique. Each cell is genetically unique. It's the same. So because of crossing over in the meiosis part, their genes are unique. Now, to summarize this one, they start diploid, and they end diploid in mitosis. But the cells that undergo meiosis, they started diploid, but they ended haploid.
Guys, I want everyone's attention. We're going to go over what we did yesterday. We're going to go over the stages of mitosis. All right, everybody ready? All right, let's go. Interphase of cell division's longest part. Nuclear membranes intact as it starts. The cell's growing, cytoplasm flowing, chromosomes get duplicated, DNA gets replicated, chromosomes are spread out so they can't be seen distinctly, but note the nucleolus, the ribosome factory outside the nucleus, the 2 centrosomes. They later make a spindle which will pull apart the chromosomes. Prophase follows, the chromosomes condense, each is made of 2 sister chromatids like an X. Each sister is a clone, the closest of kin, and a centromere connects them like Siamese twins. The nucleolus disappears and melts away as the cell takes a ribosome production holiday. The centrosomes separate, start spindle formation for separating chromatids and cell elongation. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from 1 cell to 2. Mitosis, how cells renew. In late prophase, pro-metaphase, the nuclear membrane disintegrates. The centrosomes migrate to the cell's opposing sides, and between them the fibers of the spindle wend and wind. The spindle's made of microtubule fibers which attach to chromosomes. A kinetic causes a protein patch that serves like a handle that the fibers can grasp. When they pull apart the chromosomes, splitting them in half, the spindle moves the chromosomes with nudges so fine into linear formation on the 50-yard line, a location equatorial, defined in metaphase, where the chromosomes are lined up on that middle place. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from 1 cell to 2. Mitosis, how cells renew. The spindle fibers pull on the kinetic cores. A cellular, molecular, mitotic tug of war. The centromere snaps, sisters get separated. Now these chromatids are chromosomes. They've been upgraded. The snap and separation defines anaphase. The A for apartness, for moving different ways. Kinetic core, spindle fiber separate the sisters. See, I'm waving goodbye, calling out I'm gonna miss you. And the other spindle fibers pushing grapple like felons, make the cell elliptical like a watermelon. In telophase, membranes form around the chromosomes that spread out as a nucleoli come on home. Mitosis, chromosomal ride in a pro-meta-ana-telophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. In animal cells, there's a ring of microfilaments that form at the equator, and they cinch themselves in tighter till the cell is in two pieces. Yeah, in animals, that's cytokinesis, but it's different in plants, and then the cell divides by building a new cell wall from the inside as the Golgi sends vesicles with cellulose goo, which makes a plate, then a wall divides a cell in two. And instead of one mother cell, we now have daughters too. Identical twins, kinda old but kinda new. From your single cell beginning, this is how you grew. And for single celled eukaryotes, it's reproductive too. Mitosis, chromosomal ride in a pro-meta-ana-telophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. Mitosis, chromosomal ride in a pro-meta-ana-telophase divide. Okay, guys. Eukaryotes go from one cell to two. Mitosis, all right. How cells renew.